Today we are talking about enzymes, and we are going to use packet page 36, and this is going to go on page 45 of your notebook. So this is packet page 36. It's going to go on page 45 in your notebook. We've talked a little bit about enzymes before when we were learning about proteins, but enzymes are super important proteins that help all of the chemical reactions that happen in our body, and that's why we're bothering to talk about them separately. Okay, on the very top, the first thing to know is what are enzymes? Okay, and enzymes are proteins that help speed up chemical reactions. Okay, enzymes are proteins that help speed up chemical reactions. Okay, and so make sure that we know what a chemical reaction is, right? When we look at a chemical reaction, we always start with sort of the ingredients, and we make something, we make some kind of product. Okay, so if you were if you were making dinner, okay, the ingredients that you started with, and then there would be some kind of process, and then you'd wind up with dinner on your plate. That would be the reaction, if you will. In science terms, we don't call them ingredients. Remember the fancy name for that? We call them reactants. And so our reactants are always on the left-hand side of the chemical reaction. Our products are always on the right. And when we see this arrow, okay, we can say that these make that, but the scientific way of saying it would be yields. Okay, so these reactants yield this product. So in this reaction, okay, here's my ingredients, here's my reactants, here's my arrow, okay, so that's my yields, and these are my products, water and oxygen. Okay, so that's just a little chemical reaction example there. All right, let's talk about what enzymes are, what they do, how they work. Let me make this a little bit bigger. Slide this over so you can see a little bit better. Okay, so this question here, what is a catalyst? Okay, and the reason I put this here is that you know how scientists like to use many names for the same thing. Okay, a catalyst is another name for an enzyme. Okay, so if you see in a question, if you see something about a catalyst, we're talking about an enzyme. And what an enzyme does is it lowers the activation energy of a chemical reaction. So we need to know what activation energy is. Activation energy is the energy needed to start a chemical reaction. Once it's started, then the chemical reaction will happen. But it's the energy to get it going. Okay. Enzymes speed up chemical reactions by lowering their activation energy. Okay, so it lowers the amount of energy that has to be added to make the reaction go. And here is a graph that shows that. Okay, so I want to explain this to you. Here's a chemical reaction, and it's what we're measuring is the energy level during that chemical reaction. Here's the energy level of our reactants. Here's the energy level of our products. So we can tell that we're going to go from here to there, okay? But we can't just go directly there to there. We have to add energy to make that process go, okay? So the amount of energy that you have to add, which is this bump here in the graph, that's the activation energy. How much energy do you have to add to the reactants to make them turn into the products? So this top line, this higher line, this is the amount of activation energy needed without an enzyme. It needs a lot of energy added in order to go from reactants to products. This dotted line is the graph of the lower activation energy using a catalyst. So this is with an enzyme. And what I want you to see is that the reactants don't change, the products don't change. The only difference in this without the enzyme versus with an enzyme is that you didn't have to add as much energy. So the same process happened with less energy with the enzyme than it would have without the enzyme. Okay, it's good to know some examples of enzymes. Enzymes always end in ASE. So if you see a word that ends in ACE, that's going to be an enzyme. Okay, just like carbohydrates ended in OSE most of the time, enzymes always end in ACE. Catalase, okay, is an enzyme that breaks down starch, for example. Lactase breaks down lactose. So for those of you that are lactose intolerant, your body probably doesn't make any or enough lactase to break that down, and that's why milk gives you a tummy ache. Amylase breaks down amylose, okay, which is also a sugar. Amylase is an enzyme that's in your mouth, it's in your spit, and it's why when you put like a Jolly Rancher in your mouth, like a hard candy, and it starts to dissolve, it's that enzyme that's starting to break down those sugars. That's why it literally is melting in your mouth. Okay. But be on the lookout for anything that ends in ACE. Now, the way that enzymes work is very specific. They each do a specific job. And I always think of them like puzzle pieces. Okay. And there's a name for this. It's called a lock and key model. Each protein has a specific shape. So enzymes can only bind to substrates that match its shape. Substrate is a fancy name for the protein that the enzyme will bind to, okay, or the substance rather, that the enzyme will bind to. The site on the enzyme where the substrate hooks on is called the active site. So here we see an enzyme reaction, okay? This is the substrate. And down here we have the enzyme. Notice that the enzyme doesn't change throughout the process. That's why enzymes can actually be reused after the process is done. The substrate, okay, it could be a protein, it could be a starch, it's whatever the enzyme is going to attach to, has to fit into this space, this active site. Okay, this is the active site. Active site. Right here. And so this piece, okay, has to fit into there. And so think of it like putting puzzle pieces together. If they don't fit, it doesn't go there. So enzymes will only interact with a specific substrate. They fit together, and at this point, it's called the enzyme substrate complex because they're together, locked together, and that's when the reaction will happen. And then when it's over, the enzyme will let go of the products. In this case, the substrate broke in half, okay? And then this enzyme can be reused. So the enzyme can go back and find another substrate and be used again. This active site, here's a good picture of it too. The active site and the substrate, you see how they fit together? If they don't fit together, then that enzyme can't be used with that substrate. Okay, just a couple of things to keep in mind. Enzymes can be reused. Enzymes are affected by temperature and pH. They like to be at an optimal temperature, which is their best temperature, or an optimal pH, the pH where they work best. If they get out of their temperature range or their pH range, they change shape. And there's a process, there's a name for that. It's called denature. Denature is what we call it when an enzyme breaks down or changes shape. So it would actually get almost like warped. And imagine, because it has to fit with this active site, has to fit with this substrate. If this enzyme were to denature and get bent out of shape a little bit, it wouldn't fit anymore. And so the enzymes, when they are denatured, they change shape and then they no longer fit together with their substrate and they don't work properly. That's why with temperatures, if you've ever run a real high fever, you'll notice that it's like real hard for you to eat solid food. You usually throw up because your digestive system has a lot of enzymes in it. And when you're running a fever, the temperature is higher, those enzymes denature, and they no longer are breaking down the food that they once were doing. So usually you can't, you really can't eat a steak dinner when you're running a fever because you don't have those enzymes to help you. We have in our systems, we have buffers to keep the pH stable so that enzymes don't denature. And we learned about those when we talked about the pH scale and acids and bases. But buffers are chemicals that keep the pH of the environment stable. Make sure that you put that in your notebook, and I will see you soon.